Vitamin D has long been associated with bone health, primarily for its essential role in calcium absorption and bone mineralization. But modern research has significantly broadened our understanding of this nutrient, revealing that its influence extends far beyond the skeletal system. Vitamin D functions more like a hormone than a simple vitamin, and its reach touches nearly every tissue in the body, including the immune system, cardiovascular system, brain, and even the machinery of our genetic code. One of its most profound and underappreciated roles is in supporting genomic stability, a foundational pillar of long-term health and disease prevention. At the molecular level, vitamin D exerts its effects through the vitamin D receptor, VDR, a nuclear receptor found in nearly every type of human cell. When activated by the hormone's biologically active form, calcitriol, this receptor binds to DNA and directly influences the expression of hundreds of genes. Many of these genes are involved in critical processes such as DNA repair, cell cycle control, immune response, oxidative stress regulation, and inflammation reduction. In this sense, vitamin D acts as a genomic regulator, not merely supporting physiological functions, but helping maintain the integrity of the genome itself. One key aspect of genomic stability is the protection of telomeres, the repetitive DNA sequences that cap the ends of chromosomes. These structures serve as a buffer against DNA damage during cell division, and their gradual shortening is considered a hallmark of aging. When telomeres become too short, cells enter senescence or die, contributing to tissue dysfunction and age-related disease. Vitamin D has been shown to play a protective role in telomere maintenance, likely by reducing oxidative stress and chronic inflammation, two major accelerants of telomere shortening. Several studies have observed that individuals with higher levels of vitamin D tend to have longer telomeres, suggesting that maintaining adequate levels could help preserve cellular youth and genomic integrity over time. Another way vitamin D contributes to genome protection is by enhancing the body's DNA repair capacity. It influences the expression of genes involved in nucleotide excision repair and double-strand break repair, mechanisms that help the cell fix mutations before they lead to cancer or cellular malfunction. Without sufficient vitamin D, these processes may become less efficient, increasing the risk of genomic instability and the accumulation of mutations, a key driver of aging and carcinogenesis. Beyond the genome, vitamin D also helps manage inflammation and oxidative damage, both of which can impair DNA integrity and accelerate biological aging. It downregulates pro-inflammatory cytokines like interleukin-6 and tumor necrosis factor alpha, and it supports the production of antioxidant enzymes that neutralize harmful free radicals. These effects reduce the cellular stress that wears down DNA, mitochondria, and other sensitive structures. In aging populations especially, where inflammation tends to rise and antioxidant defenses decline, maintaining adequate vitamin D levels can act as a buffer against systemic decline. The immune-modulating effects of vitamin D are also deeply relevant to genomic health. An overactive immune system, particularly one prone to chronic inflammation or autoimmunity, contributes to oxidative DNA damage and telomere shortening. Vitamin D helps balance immune responses by encouraging regulatory T-cell function and tempering excessive immune activation. This not only reduces the wear and tear on tissues, but also supports immune surveillance, the process by which the body detects and eliminates abnormal or precancerous cells. Vitamin D's influence even extends into the realm of epigenetics, the chemical modifications that determine how genes are expressed without changing the underlying DNA sequence. Some studies have shown that vitamin D can impact DNA methylation patterns, which are closely linked to biological aging. This epigenetic influence may be one more avenue through which the nutrient helps preserve the functional potential of the genome throughout life. In light of all this, it becomes clear that vitamin D is much more than a bone health supplement. While its role in preventing osteoporosis and supporting calcium balance is well established, its deeper contributions to maintaining genomic stability, regulating immune function, suppressing inflammation, and slowing the biological aging process are equally, if not more, significant. Especially in a modern world where deficiency is common due to indoor lifestyles and limited sun exposure, ensuring adequate vitamin D intake may be one of the most accessible and impactful strategies for protecting long-term health at the cellular level.